Good morning, everyone. Today I'm here to talk to you about enhancing the throughput of the ProteOn XPR36 biosensor and antibody screening applications. And as Laura mentioned, I am a scientist at Rinat Pfizer in a biosensor core lab within the protein engineering group. I'm having a little trouble with the slides. One moment. Okay. So I will focus on three different types of antibody characterization assays that we perform on the ProteOn biosensor. And for those of you who may not be familiar, the ProteOn is a real-time label-free biosensor that is SPR detection-based, and we use it for many applications. In particular, I'm going to talk about epitope bending applications, epitope mapping, and kinetics and affinity characterization of antigen-antibody interactions. And the majority of the work I'm going to show today was recently published in Analytical Biochemistry, where we go into the detail on how to increase the throughput of the ProteOn biosensor beyond what may have originally been anticipated on this platform. And in our group, we use the ProteOn, and this is actually a picture of the instrument on our lab bench in our lab. And it's a little difficult to see in the picture, but you have a plate-based autosampler where there are two 96-volt plate positions, and there's an autosampler arm that will move 6 needles over to the plate. And so the ProteOn has the ability to deliver 6 analyte injections in parallel over a sensor chip surface. And there is a unique microfluidics-based design to the ProteOn, where there is a microfluidics device that mounts on the sensor surface, creating 6 flow paths, and the analyte can be injected, say, for example, you can see in the red arrows in a horizontal direction. The instrument can undock the fluidics and rotate them 90 degrees and redock, and then you could inject 6 parallel injections in the vertical direction. So we have this set up with intersecting flow paths. And this feature allows us to create a 6x6 interaction array, and I will get into the details on how we immobilize up to 36 different ligands on this sensor chip in a few more slides. And as I mentioned, the ProteOn is SPR detection-based, and so it measures very small refractive index changes on the sensor chip surface, and these refractive index changes are directly proportional to the amount of mass that's bound to the sensor chip surface. Sorry, I'm having trouble advancing slides. So when it comes to increasing throughput on any SPR-based platform, I think it's obvious that if you can monitor more interactions simultaneously, you will increase your throughput. So that's where most people focus. And on the ProteOn, since we have these 6 parallel injection systems, we can look at up to 6 analytes. And now we have this array that we can create with up to 36 ligands. So we have a potential of 6 analytes and a sensor chip surface that could have 36 things immobilized on it. So we are fixed to that number of interactions that we can monitor simultaneously. Also, when it comes to increasing throughput, we like to minimize our hands-on setup time. So having intuitive control software, we can set up automated methods and configure things in a typical plate format to make them easy to pipette are things that we focus on to increase our effective throughput in the sense that the user can go do other things. So unattended runtime becomes very important. So this is where autosampler capacity is important, and we can set up in the ProteOn control software long automated methods that could go overnight, that could go over the weekend, and that allows us to utilize all that extra time when the operators are not in the lab, but the instrument will keep working for you. And also, as the throughput of these real-time label-free biosensors increase, we need data processing tools that can handle data for these large data sets and automate the steps for analyzing the data, for fitting the data, and the ProteOn evaluation software has options for that as well, and we use both the ProteOn evaluation software and in-house tools for analyzing these large data sets. So just a quick introduction for anyone who's not familiar with the primary data output of an SPR-based biosensor. On the y-axis, we have response units, and these are arbitrary mass units in the sense that we're measuring refractive index. That refractive index change is directly proportional to the change in mass, and we don't need to know the actual mass. Having arbitrary mass units allows us to analyze the data for kinetics and affinity parameters and so forth. So on the x-axis, we have time. And in the sensorgram, we typically have a baseline response. So you can see from about 0 to 120 seconds, there is a flat response. That's a constant mass on the sensor chip surface, buffer flowing over it. And then an analyte injection will begin. When the analyte associates with the surface, we get an increase in our response. And at the end of the injection, just past the 200-second time point here on this sensorgram, buffer flow begins to go over the surface, and we watch the dissociation of the complex. So we are generating many of these sensorgrams simultaneously on the ProteOn instrument. This unique microfluidics design for the ProteOn allows us to use various multiplex formats. And when I say multiplex, I mean multiple analytes on multiple ligands. And one of the early applications on the ProteOn that was published is one-shot kinetics, where we could immobilize up to 6 different ligands in the vertical direction as shown in the color vertical strips there on the figure. And then the fluidics can be rotated, and in parallel, one can inject a dilution series of your analytes. You would have typically, say, 1 antigen binding on these 6 different antibodies. Typically, one of those flow channels is reserved as a buffer blank. So you would have a buffer blank and 5 different dilutions of your analytes. This is a very rapid way of getting kinetics for 1 analyte on 6 different ligands. If one were doing a screening application, you could, for example, have 6 different analytes being injected over 6 different ligands. And what I'll focus on for the remainder of this presentation is a format where we would have 1 analyte being injected on 36 different covalently linked ligands on the sensor chip surface. And I'll talk about how to create that surface in a moment. And one could also actually work on unrelated projects on the same sensor chip in the same run, as shown in the bottom right-hand side of this figure, where you could have 1 analyte on 6 unique ligands and have yet another analyte on another 6 ligands in the other channel, and so on and so forth. So they could be things that aren't even related. They could be completely different projects. And I think the multiplex format you choose will depend on your application. So in antibody engineering applications, which I am often involved in, we typically have a large panel of antibodies and a small number of analytes. So perhaps an antigen or antigen from different species on the same panel of antibodies. So the 1 analyte on 36 ligand format is the optimal format for throughput for us. But if you were, say, doing small molecule protein interaction work, where you typically have a small number of proteins and a large number of analytes, something like that 6 analytes on 6 ligands format may be the optimal choice for throughput. So the flexibility is nice and allows you to configure the assay to optimize throughput for your particular application. So when we create an array of 36 different full-length IgGs, we do it in the following manner. You can tell the instrument to activate one of the horizontal channels. This is typically with EDC sulfo-NHS mixture, and that mixture, when you inject it over the sensor chip surface, will activate carboxyl groups on the sensor chip surface. You'll form an NHS ester, which becomes reactive to primary amines. So this is a standard primary amine coupling. So that's the horizontal direction in one lane, and there is buffer in the other lanes. And then the fluidics can be rotated. Ligand can be injected. 6 different ligands can be injected in parallel to immobilize them on those activated spots. And that ligand injection takes place in the vertical direction. Then those surfaces are blocked with ethanolamine, and then the fluidics are rotated back again, where this process is repeated on the second channel. And so on and so forth. With multiple rotations, we can create 36 immobilized IgGs on this array. And we also have, as you can see in that checkerboard pattern in the bottom right, we have options for the types of reference surface available to us. In the horizontal direction, we have these areas called interspots. They're shown here in purple. That is where the channel would have been activated and blocked, and there's no ligand immobilized there, so that's one type of reference surface you could have. And in the vertical direction, you have the gray areas in the interspot, which would be just bare chip. So this is the sensor matrix without any blocking. So you have a choice. Typically for protein-protein interactions, we like to use the activated and blocked interspots as our reference surface. Okay, so after we've created such an array with 36 different immobilized ligands, and as I will show you these results, we actually used 31 different antibodies and had various controls on the array and replicates and so on. This is an example of a result from one spot on the array. So when you gather this much information, it's difficult to show it all in one figure. And this is an example of a typical pairwise epitope binning strategy where we have the immobilized IgG shown in dark blue on the sensor chip surface on the right, and then we have the antigen that we inject and bind to it. So if we look on the sensorgrams on the left-hand side, we have antigen injected, immediately followed by a potentially competing antibody. So if the antibody is able to simultaneously bind antigen, we'll see a binding response in the second part of the injection. So where you see the arrow that says array of sandwiching IgGs, that's where the competing antibody injection begins. We have a self-sandwich as a negative control. So when the competing antibody is the same antibody that is immobilized on the chip, we should get no response. And you can see that we go immediately from antigen injection to antibody injection without any intervening buffer wash. And this is a co-injection feature that's available on the ProteOn instrument. And this is valuable in that if you have an antigen that rapidly dissociates from the surface for that particular antibody, we can still get information on that interaction. If you had an intervening buffer wash, perhaps the antigen would all wash away before the competing antibody has a chance to bind. And that would only happen for fairly weak affinity interactions, but it can occur sometimes, so we like to use the co-injection strategy to avoid that. And the result I'm showing you here is a result from one spot where we did multiple rounds of antigen binding and antibody binding with a regeneration step. So doing a large panel of pairwise interactions on this array requires regeneration. We also could use other binning formats on this array, such as a premix strategy, where instead of injecting just a competing antibody, you actually just inject a mixture of antigen and antibody over the mobilized IgG. So typically here, you would have a control where you have just antigen binding alone, and then mixtures of antibody and antigen injected to see if you actually get an increase in response or a decrease in response relative to antigen alone. We call that a premix strategy. And you could also do an in tandem strategy where you would inject, say, over an immobilized antigen and saturate with an antibody and then come in with another competing antibody. That format has antigen down, so the throughput there would not be quite as high as the classic sandwich or the premix format. So if we take all of the data for the entire array and the multiple rounds of binning that we do, we can actually investigate the whole array of 36 by 36 pairwise interactions. And before I show you the final result, we always like to corroborate any new application with an orthogonal technology to be sure we're getting consistent results and they agree with one another. And we have also BLI-based biosensors in our lab, and they measure a thickness on a sensor surface. As you can see in the right-hand side figure here are the same set of antibodies as are shown on the left-hand side. The left-hand side is the ProteOn data, right-hand side is the BLI data, and we get the same results regardless of the technology. On the SPR platforms, we have a better signal-to-noise ratio, so for things that give a very small response, sometimes it can be clearer on the SPR data if we actually have a sandwiching interaction or not. So when we, in our application, we have, as I said, 31 antibodies. It takes about 3 hours to create that array. We do up to 36 different co-injections to get the whole matrix here, where we have every antibody is immobilized and every antibody has a chance to be the competing antibody. So we actually get all pairwise interactions, and we also get them in both orientations. So a given antibody has a chance to be immobilized and be the competing antibody. And this allows us to investigate if there are any asymmetries in this matrix. For the data I'm showing you here with this particular engine, this particular panel of antibodies, there were no asymmetries in the matrix. So what we're showing here is green cells in this grid are those antibodies which are able to pairwise bind. The red cells are where the interactions are blocked. And the ones that say SS are self-sandwich controls, so those all should be blocked. This grid has also been sorted to group them into the different bins. So as you can see, in this particular case, we investigated 31 antibodies. We ended up with 13 different bins. Some bins are unique, such as bin A, and some bins are partially overlapping, such as bins B and C. And an important thing to note here is that in this particular case, bin H, which contains 3 antibodies, was the only one that had antibodies with the desired behavior in the cell-based assay. So those antibodies were the ones that we thought would most likely be functional. So when people are increasing throughput on biosensors, sometimes they tend to think that they're going with lower resolution data, sacrificing resolution for the throughput. But in binning, it's actually the opposite. When we have more interactions to look at, when we can look at them in both orientations, we actually get more information. We get a higher resolution binning. And in this case, looking at 31 antibodies, only 3 had the desired functionality. So being able to look at much larger matrices, we believe is an advantage in finding the antibodies that you want to move forward with. So as I mentioned, 3 hours to create this array, about a 9-hour total runtime, and we get this entire matrix. So I'll move on now to an epitope mapping application, and I would like to take a moment here to just say that we make a distinction when we say epitope mapping versus epitope binning, and in the literature you may see the terms kind of used interchangeably. But when we say binning, in my lab, we typically are referring to these pairwise tests for the ability of an antibody to simultaneously bind an antigen. Epitope mapping are those applications where we actually get information on which amino acids are involved in the binding. So in this case, we are looking at the ability of our panel of antibodies to bind to linear peptides, and those peptides are short peptides which represent the entire primary amino acid sequence of our target antigen. So we will obtain biotinylated peptides. In this particular case, we have biotinylated 15-mer peptides. They overlap by 10 amino acids each, and as I mentioned, they represent the entire amino acid sequence of our target antigen. So if we have antibodies that bind any of these linear peptides, it gives us information on the actual amino acids involved in binding. So we'll refer to these types of applications as epitope mapping. And we create this peptide array on the proteon by utilizing a mean coupling chemistry in a slightly different way. We will inject activation solution in the horizontal flow path, then inject streptavidin, and then block with ethanolamine, all in the same horizontal strip. And then rotate the fluidics and inject peptides 1 through 6. So the biotinylated peptides bind to the mobilized streptavidin, and then we block with free biotin or a free biotin conjugate, such as amine PEO biotin. We'll rotate the fluidics again, couple streptavidin by amine coupling in another horizontal channel, and so on and so forth to create an array of up to 36 different peptides. And the result that I'll show you is for an antigen that had 24 different peptides, as shown on the right-hand side of the figure. I am showing data here from 5 different antibodies, A, B, C, D, and E. And we perform this application on 3 different biosensors, the ProteOn, the Octet, which is the biolayer interferometry-based biosensor, the same one I referred to earlier in the pairwise epitope binding comparisons, and the BioSensor SPR-based biosensor. So the ProteOn format is advantageous in that you have the entire peptide library mobilized on one chip. So you simply inject the antibody that you're testing for a few minutes, watch it dissociate for a few minutes, then regenerate the sensor chip surface, and then do that with another antibody. And we get roughly 1 antibody every 10 minutes that we can map this way on the ProteOn. And we see that the results are very similar regardless of the biosensor technology that is used. The peptides here are color-coded to show which ones bind, and on the ProteOn figure in the upper left for antibody A, you can see a couple of duplicate sensograms. That's because we actually had some peptides replicated on different spots, just to show that we get the same result regardless of where the peptides are mobilized on the array. This is a very rapid way of getting this information. Also, as you can see, for antibody E, we didn't get significant binding to any of the peptides, and it's thought that this is because antibody E is very conformationally sensitive. So sometimes you do not get a mapping, but that's also additional information. And this type of information can be useful, for example, in not just the drug discovery related part of our projects, but also in hunting for reagents that we can use for tools. So those antibodies that bind linear peptides, say, might be more useful in a Western than others, than conformationally sensitive antibodies. So we have these higher throughput epitope binding and epitope mapping applications. We use those to narrow down the number of antibodies that we want to characterize in detail before we go into a detailed kinetics and affinity analysis. So when we have our panel of antibodies that we want to test further, we will typically perform a kinetics assay. And I'm showing here the standard one-shot kinetics methodology that I talked about earlier. And on the right-hand side, I'm showing a result that we have obtained in our lab with the one-shot methodology. This happens just to be one antigen-antibody interaction. But when you look at this, each curve is not generated in a serial fashion. These are all generated simultaneously. So you have a buffer blank and the 5 different analyte concentrations, those curves all being collected simultaneously. And we showed the red lines are the fit overlaid onto the data. So in one analyte injection with dissociation, we collect 6 equivalents of that figure. On the 36 ligand array, we do this slightly differently. We will inject an analyte concentration, watch the dissociation, then regenerate the chip surface, and do this again at another analyte concentration, and so on and so forth to generate the full dilution series. But when we've done that, we get 36 equivalents of that figure that you see in the bottom right-hand side. So that gives a comparison of the amount of data that one collects. And because we can take advantage of the long unattended runtime or an overnight run, we can collect kinetic information of the 36 antigen-antibody interactions in one run. We also try to save time in the assay when we can by using different techniques. And when we use different techniques, we like to compare them to more traditional techniques to be sure that we're getting the same answers. And here is an example where we perform the kinetics on the 36 ligand array, as I explained, but we did it slightly different ways. On the right-hand side is a more traditional approach where the association time, or the time that you inject the antigen, is the same for all antigen concentrations, and the dissociation time is the same for all antigen concentrations. But when you have very low antigen concentration, the dissociation phase of that sensogram doesn't actually give you much information about the dissociation rate of the interaction. It's a fairly horizontal line. So you get the most information about dissociation when you have more antigen bound to the surface. So when there's more antigen bound on the surface, you get a more response unit change over your dissociation profile. So we can do this short and long injection method strategy where for the lower analyte concentrations, we watch dissociation for a short time, and for the very highest analyte concentration, we watch for the longer time. And this allows us to save a lot of time not gathering less useful data of dissociation when there's lower amounts of antigen bound in the sensor chip. This is particularly important for these parallel systems. What's nice when you have very stable interactions and therefore need a very long dissociation time, say it takes an hour or two of dissociation, and I believe the examples I'm showing you here, it's about a half hour of dissociation time. We're collecting that in parallel on 36 different interactions. So this is where the parallel nature really helps, especially when you have long steps such as these long dissociations. It really helps the throughput. So we see here, comparing these two different methodologies, we get very similar answers for this particular interaction. And just as a note, a technical note on how these data were generated, we have, say for example, the left-hand figure, the same antibody immobilized on two different spots. We have globally fit the association and dissociation rate constant across those two spots. But each spot has its own Rmax value, so its own analyte binding capacity value, because there will be slight differences in the amount of immobilized antibody. So you can see here we obtain a good global fit across two spots. And the same methodology was employed for fitting on the right-hand side figure. Also, another thing that we do when we're doing higher throughput kinetics, we tend to use a higher dilution factor. So a 3-fold or a 5-fold dilution factor, you may see kinetics results from others where you have a 2-fold dilution series. But when you have a whole panel of antibodies, you're going to have more diverse kinetic profiles, some weaker affinity interactions, some higher affinity interactions, and having a bigger dilution factor helps. It's the concentration range that matters more when it comes to getting accurate kinetic information. So just to expand on the previous slide, I'm just showing another example of another interaction with slightly different kinetics, showing that we get very similar results either way. So there's more than one way to do these things, and we tend to do validation work to be sure that these optimizations we're making regarding throughput are not sacrificing data quality. So this slide is showing what I referred to earlier in the sense of having diverse kinetic profiles, and that the ProteOn array, this 36 ligand array, is capable of quantifying interactions with the diverse kinetics. So we have, for example, on the upper right-hand side of this figure, a more stable interaction. I believe the dissociation rate on the upper left-hand figure is a more rapid. That data has a fast on rate, a faster off rate, is partially mass transport limited, but we still can fit that data well and get the kinetics and affinity for that. We also can get affinity on low capacity surfaces, such as the spot on the bottom right-hand side of the figure. So that is around, I think, 20 response units for the Rmax on that surface. And here, just to make a note, we have the notation of, say, L1A3 in the ProteOn control software. The direction of the horizontal and vertical is referred to as the analyte direction and the ligand direction. We like to call them horizontal and vertical in our lab because sometimes when we inject analyte, we're injecting it in the ligand direction, and this is just the nomenclature inside the ProteOn software. So if you're looking at the software, it's called ligand and analyte direction. I like to refer to them as horizontal and vertical. And also I show here the results on the lower left-hand side where we have a blank spot, where basically nothing was coupled to that sensor chip surface. It was just activated and blocked, and we can see we get a nice negative result. So we tend to include positive and negative controls on these arrays just to validate that everything's working properly. And also, sometimes we will get interactions that don't yield kinetic information. These tend to be weaker interactions with faster on and faster off rates. And when that occurs, the sensograms come to equilibrium rapidly. So we can look at the equilibrium binding response levels and plot them versus concentration and get the equilibrium dissociation constant. So we lack the kinetic information, but we still get affinity information. And we've also done this in a 36 ligand array format for large panels of weaker interactions. And the results on the left-hand side there are showing results of multiple analyte concentrations in duplicate, so we get nice reproducibility. So the ProteOn XPR36 enables higher throughput antibody characterization assays, and I showed you examples in epitope binding, epitope mapping, and kinetic characterization. And expanding from, say, the one-shot kinetics methodology where you have 6 ligands immobilized using this 36 ligand array, and the unattended runtime that the instrument enables allows us to investigate larger numbers of interactions simultaneously, larger numbers of interactions in an unattended run, helping us in our daily work in performing antibody-based drug discovery. So with that, I would like to conclude my presentation and open it up for questions. Oh, and before I forget, I always would like to thank everybody who makes this work possible. Yasmina Abdish is the director of the Biosensor Core Lab here at Rinat, and Alana Pinkerton is a research associate in our lab that has helped with much of this work. Arvind Rajpal is the director of the Protein Engineering Group at Rinat, and Jaume Pons is a VP of Research at Pfizer-Rinat, and they all make this work possible. Jaume and Arvind support our group very well and give us these nice instruments to use. So with that, I'd now like to open it up for questions.